Okay, and last but not least, we're going to talk about how the nervous system accomplishes all the jobs that we've already spoke about it doing. So we talked about the central nervous system being composed mostly of interneurons, which interpret the information that they receive from the sensory neurons, which collect the information and then send the signal down to tell the motor neurons what they should tell their effector cells to do. Now we're going to look more closely at neurons and how they send these signals, how they actually get this information across to each other and to their effector cells, their cells that actually do the jobs. So we looked at the anatomy of a neuron 2 videos ago. So we know that it consists of a cell body, which is what we see here in this picture. This is the cell body. As well as an axon along which the signal will actually be sent. And then the axon ends in terminals. And this is what this is a big picture of. You see, this is a big blown up picture of an axon terminal. And so we're going to talk about how neurons actually collect the information, decide whether or not to send a signal, and then send a signal. And this is all done electrically. So we're going to be talking about electrical impulses within the cell and then chemicals between the cells. So we'll address all of that throughout this video. So basically what happens is the cell body, like we said, is the place where we interpret information, and there are what we call afferent fibers or dendrites. Afferent just means moving toward the cell body, and they're going to bring that information into the cell body. So here we see some information coming in that is positive. Here we see some information coming in that is negative. Basically, the positive is encouraging the cell to send a signal. The negative is discouraging the cell from sending a signal. And the cell body's job is to decide which one wins. And in this particular picture, we see there's more positive influence than there is negative influence. And so that may cause the cell to send a signal, to go ahead and send that signal down the axon. This particular location here is called the axon hillock, the very beginning of the axon. And that's where the process of summation, or bringing all that information together and adding it up, happens. Once there is enough positive influence here, then that signal will be sent down the efferent fiber, which means moving away, and that's the axon. So it's going to send that signal down the axon. In the early 1900s, when scientists were studying the nervous system and really learning a lot about how the nervous system worked, they were really surprised, because what they found out was that at the end of the axon, when a signal was sent down the axon, at the axon terminal, there was this space. The axons didn't actually touch their effector cells. Now, if that's a sensory nerve or an interneuron, their effector cell is going to be another neuron. But if it's a motor neuron, then that effector cell is going to be a muscle. But in any case, they don't actually touch the cell that they're having to communicate. There's a gap. And that gap is called the synapse. They called it that space where they come together, but they don't actually touch. And that led to the discovery of how nerves actually send signals to other cells, which is through the use of chemicals called neurotransmitters. Now, neuro just means nerve, and transmitter we know means to send a signal. So these are signal-sending chemicals that are actually sent out through exocytosis. And you remember exocytosis is just the use of vesicles fusing with the membrane to send out chemicals. So we can see these vesicles here fusing with the membrane and the chemicals being released. Those chemicals are recycled through what are called reuptake pumps or reuptake channels, so that they cause the change in the cell that they need to cause. They jump the synapse, they move across that gap, and then they are recycled. They're brought back into the cell so that they can be used again, which is really efficient of our body. So to sort of sum up how the nerves get their information, the information comes from other nerves, usually sensory nerves, or in the case of a sensory nerve, it gets its information from the outside world. They're specially adapted to either respond to light, pressure, heat, sound, whatever sensory organ they're involved in. And they get that information in the cell body. Some of that information tells the cell to send a signal. Some tells it not to send a signal. When there's enough information saying, yes, you should send a signal, that it overwhelms the don't send the signal message, then that signal is sent down the axon. It travels down the axon, and then it causes the release of neurotransmitters into the synapse, that space between the nerve cell, the neuron, and its effector cell. So we can take a look at some neurotransmitters. Like we said, they're chemical messages from a neuron to its receptor cell. What its receptor cell is depends on what kind of neuron it is. So if it's a sensory neuron, its effector cell is going to be another nerve. If it's a motor neuron, then its effector cell will be a muscle. You've probably heard of some neurotransmitters: adrenaline, which we mentioned in the last video, dopamine, serotonin. These are all natural neurotransmitters that our body uses to cause different communications between cells. So these are all examples of neurotransmitters. And you can see sort of the process of a neurotransmitter in action in this picture here. Those neurotransmitters are going to go into the next cell, and they're going to cause a change in the membrane of that next cell. And that change can either make it more likely that something happens, so it can be a positive change, or it can make it less likely that something's going to happen. So it can close something off. It can make it less likely for that effector cell to contract or to send another signal. So they can be what we call either activating, which would be positive, or inhibitory, which would be the more negative effect. And so depending on what neurotransmitter it is, that will determine the effect that it has. Interestingly, since we've discovered neurotransmitters, we've discovered that a lot of drugs and medicines, some synthetic chemicals, act like neurotransmitters or interfere with neurotransmitters. And this is just a picture. You don't have to know everything that's in this picture, not by a long shot. But it's just a picture that gives you some indication of things that act like neurotransmitters or interfere with neurotransmitters. So if we look here, you can see black widow spider venom. Is here, right? It's a very powerful venom. It actually interferes with the action of neurotransmitters. It stimulates the release, so it causes our nerves to have more neurotransmitter release than it's supposed to be, basically firing our nerves when they're not supposed to fire, and that causes the symptoms that come along with black widow spider venom, usually death if is left untreated. Botulinum toxin, which is one of the most toxic substances on Earth, also interferes with neurotransmitters. It inhibits the release of neurotransmitters, so it basically stops our neurons from being able to send signals. You see a couple other things on here that you're familiar with, probably nicotine, which is found in cigarettes, cocaine, which is obviously a very dangerous drug. They mess with these neurotransmitters, and that's how they cause the effects that they do. And so a lot of drugs and medicines act at the synapse. They interfere with the natural process of neurotransmitters. So they either stimulate neurotransmitters or inhibit neurotransmitters, but either way, they're going to make the nervous system not work the way that it's supposed to. And that's one of the dangers of too much medication or drugs in general. And so let's take a closer look now at how that signal is actually sent down the axon to cause these chemicals to be released into the synapse, that space between them. So before these vesicles will fuse with the membrane and release out, like we see happening in this picture, there has to be a signal sent from the cell body down to the axon terminal, and that is an electrical signal. And so we need to look at what's going on in a neuron to see how can an electrical signal travel down the neuron, because it's not like a wire. It's not like a copper wire where the electrons can just move down. But it's all about ions. It all comes down to the ions that are in the cell, because electricity really isn't just about electrons. It's about charged particles, and ions are charged particles. Electrons are also charged particles, which is why they're used in electricity most of the time, but we don't have the sort of wiring in our body to do that. So we do it with ions, and these are actually positively charged ions. So you remember that we studied the sodium-potassium pump a while ago. And so when a neuron's not sending a signal, when it's just hanging out at rest, doing its thing, the sodium-potassium pump is active. And if you recall, it's going to send 3 sodium out and 2 sodium in. Sorry, 2 potassium in. 3 sodium out, 2 potassium in, so that you get a lot of positives on the outside of the cell and less positives on the inside of the cell, which makes the inside of the cell negatively charged relative to the outside, right? It's not that it has an overall negative charge, although there are a lot of negative ions also inside of the cell. So that overall, relative to the outside, it's more negative inside and more positive outside. Well, that difference in charge is measured in volts. For the cell, it's measured in millivolts because it's such a small difference. And it makes the inside of the cell more negative, and the typical negative sort of measurement of that is about negative 70 millivolts. So a neuron that's just sitting there, not sending a signal, not doing any work, has a difference in charge between the inside and the outside, measured at negative 70 millivolts because of the sodium-potassium pump, because the sodium-potassium pump is sending 3 positives out, but only 2 positives in. So that makes the outside more positive than the inside. Well, one of the things that neurons have that other cells don't have as many of, which makes them able to send an electrical signal, is that they have other ion channels that open up. They're called voltage-gated channels. And they open up if that potential, if that difference in charge changes from negative 70 millivolts. We call that negative 70 millivolts resting potential because it's when the nerve is at rest. That's how much difference in charge there is. Potential just means difference in charge here. It's not like it is the potential to do work. It's how much electricity it could potentially have if it were connected by like a wire or something, if those ions were allowed to move. So resting potential is negative 70 millivolts. And what we have in these neurons that's different from a lot of other cells are a lot of what are called voltage-gated channels, which means if that number changes, if that negative 70 millivolts changes, they will open up and allow ions to move one way or another. And that's how the signal actually gets sent. The signal that is sent down a neuron is called an action potential. So this is the signal that is going down the axon. It is called an action potential. This is the nerve signal. And it's all about what we call polarization, or where the positive and negative is. So at rest, we said that the outside of the cell is more positive and the inside of the cell is more negative, based on the charges on either side of the membrane. We said that resting potential is negative 70 millivolts. Well, if for some reason that changes and gets up to what we call threshold, and threshold is just the amount that you have to reach before a signal is sent, at negative 55 millivolts, if that negative 70 millivolts gets up to negative 55 millivolts, then an action potential will be fired. So this is threshold. If you get to threshold, an action potential will happen. And remember, we said that nerves are all or none things. So if we send a signal, we are sending a full signal. It's going, and it's going all the way. And this is all controlled by ion channels. So how would we get a change from negative 70 millivolts to negative 55 millivolts? Well, what that means is that we've gotten less negative, right? The inside of the cell has gotten less negative than the outside. So what must have happened? Well, some positive ions must have made their way into the cell. And that's what the signals do when they come into the cell body. The signals from other nerves communicating with that cell are going to open up or close ion channels. And if they open them up, then some positive ions will get in and will sort of raise up this potential. It'll make it get higher, but then it might fall down if there's not enough or if other things close it off. But if enough of them open up and we get to negative 55, that's when we start an action potential, which we will talk about in our next video.